welcome to 16.3. Chapter 16 has been about replication, but this specific section is going to be about DNA and the packaging of DNA. And so when we think about DNA packaging, it should bring to mind the concept of protein structure, the four levels of protein structure or protein folding. And so with DNA packaging, at every level, the chromosome is going to get a little bit wider and a little bit wider. So notice as we move forward that it's at each level the chromosome is going to just take on a greater diameter. And so at the very beginning, or the very far left of this picture, you can see the ribbon model of our DNA. And this is going to have, because phosphates are negatively charged, and we have phosphates, a phosphate sugar and a base, in a nucleotide, it is going to be a negatively charged molecule. And so the first step is the addition of histones. Histones are positively charged, and so they are going to be attracted to this negatively charged molecule of DNA. And here, at this level of packaging, our molecule is 2 nanometers in diameter. As we move forward, we're going to move up and we're going to be at 10 nanometers in diameter. So the second step is taking those histones and putting them into packages of 8. You can see those right here, packages of 8 histones. And so those 8 histones are going to come together and create a nucleosome. And so nucleosomes are used to help the packaging of DNA, to help it coil up. And so the DNA wraps around each nucleosome twice, and the DNA in between, right here, this is called linker DNA, so in between the two nucleosomes. When the DNA is wrapped around nucleosomes, that brings our width of fiber to be about 10 nanometers. And remember that the tails are pointing outward for these histones. And when we talk about the process of replication and transcription, we don't talk about this, but it does require the removal of these nucleosomes. So there are enzymes that are working to help do this, but we do not consider that in our study at this level. The third level of packaging, we are at 30 nanometers. And so what's happening here is that the nucleosomes are going to sort of wrap around coils. And it is the positively charged tails of the histones that are attracted to the DNA that help it to fold over. And you can see that we are right around here. And so we are at 30 nanometer fibers here. And so it is the intermolecular forces between the histones and the negatively charged DNA that is creating these coils. And so this level of packaging is present during interphase. So this is where we are through 90% of the cell's life. The fourth level of packaging, we are at 300 nanometers, so we are getting pretty wide. So the 30 nanometer fibers coil once again into looped domains. So here you can see the 30 nanometers, and if you look, they are wrapping around in a bigger circle, which is 300 nanometers wide. I like to take a piece of yarn and spin it, and you can actually see this fourth level of packaging if you take that thread, if you take that piece of yarn and keep spinning it or keep twisting it. And so now here the fiber is 300 nanometers wide. And our next step is going to be 700 nanometers. And the folding at this point, the looped domains coil, and the coiling at this point is not very well understood. But this is where chromosomes are. This is their state when they are in metaphase, and it is about 700 nanometers wide. the packaging process is highly specific. So if there is a gene in one spot, when the DNA coils up again in the next round of metaphase, when it has divided, it is going to be in the exact same spot. So every time that the chromosomes coil, the genes are in the same place. It is highly specific. And so there's going to be different parts to our chromosomes, and there are two types of there's two states that our chromosomes take. One is heterochromatin, and the other is euchromatin. And so the heterochromatin is tightly wound, and that's going to be here. That makes it harder for it to access. The ends, the telomeres, are going to be tightly wound, and that makes sense, right? Because we don't need to read information at these ends. So it's okay that it's tightly wound here. And if you remember, at the centromere in the middle, it's repeating units. And so it's not accessed for gene expression. And so this is also highly coiled and heterochromatin. So these points here on the chromosomes where it's in purple, euchromatin. So this is going to be less coiled, easier to access. And I like to remember this. Hetero, H, is harder to read. Hetero is harder to read. EU, easier to read. The euchromatin is easier to read. And so the methyls and acetyls, these exist in the nucleus. They attach to the histones. And these are going to make the chromosomes condense. And if the chromosomes condense, it makes it harder to read. So you're going to have more methyls and more acetyls in these heterochromatin areas. And this is one way that a cell will regulate gene expression. And we're going to continue talking about gene expression in later chapters. But remember that some parts of the chromosome have junk DNA. And so these parts are going to have these higher or a greater volume of methyls and acetyls. And it's going to make it coil more. I hope this helped, and have a great day.